Welcome to Section 3 of Microbiology Fundamentals. In this section, we'll be discussing bacterial genetics. Let's get started. Here's the table outlining bacterial genetics and the various changes that can occur. The main reason you need to understand this topic is because genetic change can increase the virulence of bacteria, as you can see with this far right column. Now, virulence was covered in detail within Section 2, the previous video, but for the purposes of this lecture, just remember that virulence is anything that helps the bacteria invade the host, evade immune defenses, or inflict disease. With that framework, let's describe the first method of genetic change, random mutation. This is a vertical change, meaning that one bacterium changes randomly and then passes that change on to its progeny. This image demonstrates random mutation. Here is the bacterium with its bacterial chromosome. And binary fission occurs, that's how bacteria replicate, and one of the daughters randomly mutates part of its genome, as you can see right here, with this red part of the chromosome. This change will then be passed on to the progeny, which are represented by these two daughter cells. Going back to the table, notice that this change is slow. It takes time to randomly mutate, especially in a way that is beneficial to the bacteria. It starts slow, making only minor modifications to the genome. Over time, the genome can begin to accumulate enough changes that the bacteria can increase virulence, as we've shown over here, or even give the bacteria antibiotic resistance. As a side note, antibiotic resistance can really be lumped into this increased virulence idea. So you might think that mentioning antibiotic resistance is kind of redundant in this context, but it's actually really important to recognize antibiotic resistance itself, so I thought I'd give it special mention here. Now let's talk about phase and antigenic variation. Just like random mutation, this is a vertical change in that the changes are passed on to progeny. However, unlike random mutation, phase and antigenic variation is very fast. With phase and antigenic variation, genes are not mutated. They are just randomly turned on or off. This random alteration can change the antigenic structure of the bacteria, which can help it evade the immune system, or it can change the structure of the bacteria. For example, by changing the capsule or adding or taking away a capsule or losing pili. And of course, these changes are significant because they can increase the virulence of the bacteria. This image depicts phase variation. First, focus on this bacterium in the middle, and we can see 4 genes. 1, 2, 3, 4. In this example, the orange gene is responsible for the pili, and you can see a pilus right here. The purple genes are responsible for some of the antigens on its surface, and you can see the antigen right here. Now let's look at the left image, following this arrow. You can see that the gene for the pili, the orange gene, is turned off, and so the pilus is absent. This is a totally random alteration. However, if this change increases the virulence of the bacteria, then the progeny of this more virulent strain will be better suited for survival, and they will keep those genes turned off. Now, if we follow the right arrow and look at this far right image, you can see one of the antigen genes is turned off right here. This changes the structure of the antigen, and you can see how it's different now. If the host immune system doesn't recognize this new antigen, then this bacterium can evade the immune system. Once again, if this change is beneficial, then it will be passed on to the progeny, and the progeny will keep this gene turned off. Now let's talk about transformation. This is a horizontal change. This means that the change is passed to nearby bacteria. This is different from vertical change, in which change is passed on to progeny. However, once a bacterium receives the genetic alteration through a horizontal change, such as transformation, it can pass it on to its progeny, which would be vertical. But it's important to know that the alteration or the genetic change didn't come from its predecessor. In other words, the direction of change is horizontal, even if it gets passed on to its progeny afterward. Transformation is a fast process, and this will make sense why in a minute. It's one of the simplest modes of change because there's simply naked bacterial DNA, and that enters the cytoplasm of competent bacteria. And this idea of naked bacterial DNA just refers to a lysed bacterium that now has its DNA free-floating in the environment around other bacteria. Now, this term competent is important because only competent bacteria can take up this bacterial DNA. There are several competent bacteria. The most important ones to remember are Strep pneumo, Neisseria meningitidis, and H. flu type B. And once inside the cytoplasm, the donor DNA from that naked lysed cell is then incorporated into the receiving bacterial chromosome. This is significant because the receiving bacterium can increase its virulence by getting that potentially beneficial DNA. Also, it's important to note that because this process is so simple, we can actually perform it in a laboratory setting. So, transformation is often utilized with laboratory research. This image depicts transformation. On the right, we show a lysed bacterium, which leads to that naked DNA. It's just floating around there. Then, on the left, we see a competent bacterium, which can then receive this DNA. The DNA then joins up with this chromosome, and now we have a new chromosome. And again, these new genes might be beneficial to the new cell, which can increase its virulence. Now let's talk about conjugation. This is also a horizontal transfer, and it's fast. And there are two types of bacteria in this conjugation relationship, the F+ and the F-. Now the notes of this process are described in detail here, and they're here for your reference, but it's easiest to show these graphically using a diagram. So here's a diagram showing conjugation. Each row shows a variation of this process. We've got the top row, second row, and then the bottom or third row. Let's start with the top row. Here's the F+ cell, and here's the F- cell. The F+ cell has a plasmid that encodes genes for the sex pilus. This is the plasmid, and it created this sex pilus. And since it could create this sex pilus, plasmids can also be called fertility factor, which is where that F comes from. In any case, the F+ cell will create the sex pilus, and then the plasmid is copied and then transferred, which you can see in the middle. Now that the recipient cell has a copy of the plasmid, it's termed an F+ cell, because now that it has that fertility factor or plasmid, it can initiate conjugation. However, something else can happen, which we will discuss with the second row. If we follow this arrow down, we can see that the plasmid has incorporated into the bacterial chromosome. Now it's got some blue and some red. If this happens, the F+ cell is now called an HFR cell. HFR stands for high frequency of recombination. In other words, this cell can create lots of new combinations of genes. Let me show you why it would create new combinations. First, recognize that plasmid genes are still present and are able to initiate conjugation. So if we follow this arrow to this next idea, we can see that there's a transfer of part of the chromosome. We've created a sex pilus just like before, and the genes are copied and transferred over to this F- cell. Now the recipient cell has these new genes in a new combination, so it's called a recombinant F- cell. Notice that it has the blue from the original bacterial chromosome, and then the red part from the donor plasmid, and of course the yellow it started with. Anyways, it's important to recognize that the recipient typically doesn't have the ability to excise those plasmid genes and initiate conjugation. Therefore, it remains this F- cell. These new genes right here may confer an advantage and increase virulence, which of course can be passed on to its progeny, but it's still an F- cell, so it's not going to transfer those genes and that benefit horizontally. It would transfer them vertically. But again, this HFR cell can still keep excising these genes and keep forming these new combinations and transferring it to new cells. Now let's discuss the third row. The HFR cell can actually excise its plasmid, which you can see described here. And when it does that, it can actually take with it some flanking genes. And you can see that blue portion that it took from the chromosome. And just like plasmids typically do, they can copy themselves and initiate a sex pilus right here. And it can be transferred to a new bacterium. And when a bacterium receives a plasmid like this from an HFR cell, it's termed an F' cell. And this F' cell has recombined genes, and those can be transferred horizontally, as though it were an F+ cell or an HFR cell. Now that's a lot of information, so let's break down the key takeaways that will help you dominate your exams. First, plasmids can facilitate new gene combinations. Second, plasmids and novel gene combinations can be transferred horizontally to new bacteria. So when you're done with this lecture and you're trying to remember conjugation, just think to yourself, plasmids. Conjugation, plasmids. So now let's come back to the table, and let me ask you this. Why would conjugation and the plasmids be significant? Well, it's because they can increase virulence, and they can even provide antibiotic resistance. For example, genes giving resistance to tetracyclines, chloramphenicol, fluoroquinolones, and vancomycin are encoded in plasmids. So bacteria can conjugate and quickly transfer horizontally these antibiotic resistance genes. Now let's talk about transduction. As you can see, there are two types of transduction: generalized transduction and specialized transduction. And these notes are here for your reference, but let's discuss these ideas through a diagram, starting with generalized transduction. In generalized transduction, a bacteriophage, which you can see right here, will inject its DNA into the cytoplasm of a bacteria. And just so you know, the term bacteriophage just refers to a virus that can infect bacteria. Anyways, this DNA enters the bacteria, and like viruses typically do, it will take over the replication machinery of its host and create new virions. In the process, the DNA of the bacterial chromosome is broken up, which you can see here. Next, the virus packages new virions into little capsids, which you can see here. And notice that some of the bacterial DNA, represented in yellow, will be accidentally incorporated into these capsids. Eventually, the bacterium lyses, and it releases those virions, or the bacteriophage progeny. These little bacteriophages can then go and infect other bacteria, and they can transfer their DNA to the next host that it attacks. And this process ultimately results in lysis of the bacteria. So this is called the lytic cycle. Lytic for lysis. So again, generalized transduction is horizontal, fast, and can be significant because it can increase the virulence of recipient bacteria. But generalized transduction is not as significant as specialized transduction. Let me show you why. This image demonstrates specialized transduction. Just like before, a bacteriophage will insert the viral DNA and it will be incorporated into bacterial DNA. And you can see this incorporation here with that yellow viral DNA incorporated into that blue bacterial chromosome. And at this point, the bacteria experiences what's called lysogenic conversion. In lysogenic conversion, the bacteria will use these newly acquired genes. Some of these genes can encode for exotoxins, which, as we've discussed in the previous video, can increase the virulence of the bacteria because they inflict disease on the host. In fact, there are several exotoxins produced from this lysogenic conversion, and we've listed them here on the right. These exotoxins include the strep pyogenes urethrogenetic toxin, the botulinum toxin, the cholera toxin, the diphtheria toxin, and the Shiga toxin. So you can see that with lysogenic conversion, the bacteria can become more virulent and release those exotoxins. However, just like with the lytic cycle we discussed with generalized transduction, these lysogenized bacteria can eventually experience lysis as well. And there's usually some type of trigger that activates the viral genes. And once that trigger happens, the bacterial DNA will then be incorporated into little viral capsids as part of the replication process. Notice that the genetic material within these capsids have some yellow and some blue, indicating that they are a combination of bacterial and viral DNA. After the formation of these capsids, the bacterium lyses, and it releases new bacteriophage progeny. And these little virions can go and infect other new bacteria. And this bacteria can be lysogenized, and then it can produce exotoxins. Now we've covered the material in this table. But before we leave the table, I want to draw your attention back to these bottom two rows. We just discussed generalized and specialized transduction. And when you think about it, viruses can really take advantage of bacteria and ultimately kill them as new virions are formed. And to combat this, many bacteria have a mechanism called restriction modification. Let's discuss this concept on a new slide. To understand the restriction modification system, you need to realize that bacteria produce DNAases, or deoxyribonucleotidases. Included in this large group of enzymes are restriction endonucleases. These restriction endonucleases are the tool of the restriction modification system. These enzymes will break down the DNA of invading viruses, and the bacteria does this to prevent the viral DNA from taking over its machinery, for example, by incorporating into its own genome. However, having these restriction endonucleases around can be dangerous because those enzymes can also break down the DNA of the bacterial chromosome. To prevent this from happening, bacteria will methylate parts of their own DNA. These are known as restriction sites, which is where the term comes from. Anyways, if methylation fails, the bacteria can destroy themselves. This diagram depicts the restriction modification system. The top row shows it working correctly, and the bottom row shows it failing. Let's focus on the top first. You can see that this virus has injected its DNA. The receiving bacteria has its restriction endonuclease, or DNAases, right here. To protect itself from its own enzyme, it methylates its chromosome at certain sites. You can see that methylase at work here. The end result is degraded viral DNA, which you can see here, but an intact bacterial chromosome. Now let's talk about when this fails by looking at the bottom row. As you can see, there's no methylase here. So the DNAase will degrade the viral DNA, but will also degrade the bacterial DNA. The end result is degraded DNA all around, which you can see all through here. So the bacteria killed itself. Now that we've discussed how the restriction modification system applies to these two items, let's discuss another item that's related to many items in this slide, especially conjugation. And this topic is transposable elements, also called transposons. Let's discuss this on a new slide. Transposable elements are simply DNA segments that can excise themselves and integrate somewhere else. For example, genes from a plasmid can integrate into the bacterial chromosome. Or genes from the chromosome can integrate into the plasmid. Or genes from one part of the bacterial chromosome can then integrate into another part of the chromosome. So what's the point of this? Well, these transposable elements can keep jumping around, ultimately being transferred from one bacteria to another. So let's think about conjugation, for example. Conjugation relies on plasmids. And when you think of transposable elements, you can imagine that a single plasmid can receive many novel genes. So these transposons integrate into a plasmid. And this can be a bad thing for us humans because it means that a single plasmid can contain antibiotic resistance genes. Not just one, but several. This is a scary thought. We'd end up having a plasmid that looks like this. And imagine if each one of those transposons carried an antibiotic resistance gene. That's terrifying. So what's the significance of transposable elements? Well, they can create plasmids that are resistant to multiple drugs. This whole table discusses how bacteria can exchange genes to increase their virulence. Now let's talk about the opposite, how they can lose virulence. Central to increasing virulence are selective pressures. Selective pressures will favor bacteria with advantageous genes, so they will become more abundant. This will increase their virulence. That's the crux of everything we discussed on that table. However, the opposite is also true. A lack of selective pressures can lead to genes, for example, plasmids, especially plasmids, that are actually not advantageous because there's no reason to have them around. So the bacteria can lose those plasmids and ultimately decrease virulence. So let's say we have bacteria right here, and it's resistant to fluoroquinolones, and it has a chromosome, and then it's got a little plasmid. And the genes for resistance to fluoroquinolones are going to be yellow on this plasmid. So we'll make this really clear. Now let's say we've got a medium here, and this culture medium does not have any fluoroquinolones. The bacteria can then be placed on this and allowed to grow on this culture medium for a set amount of time. After that time, you can examine all the DNA. So over here on the right, we've demonstrated that same culture medium after that set amount of time. At that point, you can go and look at all these little bacteria and look at their genes. And you may find that they don't have genes to give them resistance to fluoroquinolones anymore. For example, all you see is that original bacterial chromosome. So these bacteria have lost their plasmid and their resistance genes because there was no advantage to having that plasmid around or those resistance genes in this culture medium in which there were no fluoroquinolones. So the point is this: selective pressures can increase virulence and lack of selective pressures can decrease virulence, which often looks like a loss of plasmids. To dominate step 1, you need to be familiar with each type of genetic change listed on this table. For example, a question may describe one of these processes, and you will be expected to know which one is being described. The answer choices may be what you see in this column, listing the type, or the answer choices may be some detail from the description in this column, and you simply need to pick out the true statement that pertains to that mode of genetic change described in the question stem. To help you be ready for this, let's do one last question to apply what you've learned. A colony of F- bacteria is introduced to a novel culture medium in the absence of other competing bacteria. After a period of time, the bacteria is examined from 2 different areas of the culture medium. The bacteria from one area, A, has produced a slime layer of polysaccharides that was not initially present. Bacteria from the second area, B, has not produced this slime layer and are less abundant than the bacteria in area B. The researchers conclude that the slime layer was initially produced by one bacterium in the first generation. Which of the following processes describes how bacteria in area A began producing the slime layer? A, transformation, B, specialized transduction, C, phase variation, D, conjugation, or E, random mutation? Hopefully from the question stem, you notice that the bacteria in area A produced the slime layer, which gave it an advantage. And we know that it was advantageous because the bacteria in area A were more abundant than the bacteria in area B. You could say that this made the bacteria more virulent, at least as it relates to survival and growth on this novel medium. The next thing you may have noticed is that this change occurred in one bacterium, because the slime layer was initially produced by one bacterium in the first generation. In other words, we've got this culture medium, and a bacteria is placed in it. This is the first generation, and it didn't have a slime layer at first, and that's explicitly stated in the question stem. The slime layer of polysaccharides was not initially present. But one of these bacteria within this first generation produces it, and it gave the bacteria an advantage. So these bacteria are termed group A, and the bacteria in group B did not have that slime layer, and the bacteria in group A were more abundant because of that slime layer. So backing up, this first generation bacterium didn't have the slime layer, entered the culture medium, then produced it, and then it passed it on to its progeny and became more abundant. The bacteria in B, also part of the first generation, did not produce this slime layer and are less abundant. With that in mind, what explains this change? That would be answer choice C, phase variation. Recall that phase variation can change large things very quickly by randomly turning on or off genes. In this image, we depict alterations to antigens and pili. However, a slime layer would be very similar to this idea. Genes for the glycocalyx can be turned on or off, assuming, of course, that the bacterial species in question actually has the genes for a slime layer. Now choice A is wrong because transformation describes the DNA of one lysed bacteria being taken up by another competent bacteria. And in this question, we are told that there are no other bacterial species besides the one being studied. We're told there's an absence of other competing bacteria, so transformation doesn't make sense. Now choice B is wrong because specialized transduction requires a virus to transfer DNA. There's no indication that a bacteriophage was present in the culture medium. And choice D is wrong because we're told that a single bacterium gave rise to all the bacteria on the culture medium. This means that there are no other bacteria from which to gain the genes for the slime layer. Lastly, choice E is wrong because this occurs over time. It doesn't happen within one bacteria, which is what we're specifically told occurred with the slime layer. The slime layer was initially produced by one bacterium in the first generation. So one bacterium had it, and then it all of a sudden grew it. That is not consistent with random mutation over time. So again, the correct answer choice is C, phase variation.